As most of us are aware, targeting very specific signaling pathways in cancer has become a very hot area of research, with now many examples of drugs being approved by regulatory authorities. One of those pathways that appears to be very critical across all of cancer medicine is the PI3 kinase signaling pathway, which has impact on many downstream transcription factors which are known to be associated with various types of cancers, and in fact maybe even drivers in certain types of malignancies. To date, we have one drug targeting the PI3 kinase in the form of idelalisib, and there are at least 3 other drugs being developed in this space. One other well-known PI3 kinase inhibitor that looks structurally to be very, very similar to idelalisib is duvelisib, or ipi145. Another compound which has a different chemical structure, markedly different from that of idelalisib and duvelisib, is TGR-1202. It's probably the lesser known of the 3 agents. These, today I'll be reporting on some early phase 1 clinical data that seems to demonstrate that this compound has a completely different spectrum of adverse side effects and toxicity in comparison to the other 2 agents. In particular, we don't seem to see the same increase in liver function test abnormalities like we see with idelalisib and duvelisib. And importantly, we don't see the same degree of grade 3 or 4 diarrhea, and so far have not seen colitis on this particular study. The efficacy, though, seems to be right in line with what we're seeing with the other drugs. So I think it's still early, and I think we need to be very cautious because we recognize that many of the toxicities, in particular the gastrointestinal toxicities of idelalisib, occur late. And so this study is early. We are beyond a median follow-up of 6 months, which is sort of the magic number. But I still think we need to be cautious and evaluate those toxicities now over time, just to make sure that it's not a late event and all of a sudden starts to pop up. But it seems to have very good tolerability with roughly equivalent efficacy in the same types of lymphoproliferative neoplasms that we've seen with idelalisib and duvelisib. So right now, the company that's developing the drug has some registration-directed studies that have been approved under SPA with the FDA, and they're being done in CLL. There are future regulatory studies being planned in other B-cell lymphoproliferative disorders with ibrutinib. The sponsor also has a anti-CD20 that seems to have much better ADCC activity compared to Rituxan, probably on par with what we see with obinutuzumab. So at the moment, some of the first studies that are being launched in terms of combination studies are those that are being done with anti-CD20 type molecules, including rituximab and ublituximab, a next-generation glycoengineered anti-CD20 monoclonal antibody with better ADCC. And recently, George Dang from our group at Columbia, he'll be presenting a very interesting oral presentation at ASH later this year on some very interesting combination studies. I'm not at liberty to go into too much detail, but he'll be presenting some very interesting data on combination studies with other drugs that have activity in this particular space. And he can actually demonstrate that there may be some unique influences of the TGR PI3 kinase inhibitor on that pathway that are somewhat unique to the compound and may account for its complementary activity with some of these other agents on PI3 signaling that we don't see the same types of synergy with idelalisib or duvelisib. So there may be some other unique features that are not off-target effects, but in fact on-target effects if we view it from the point of view of the PI3 kinase signaling pathway. The take-home message is that there are now a number of very good, highly effective small molecules targeting these very specific signaling pathways across all of cancer, but in particular in the lymphoproliferative disorders. I think targeting the PI3 kinase pathway is going to be a very, very powerful way to produce responses in patients that may be resistant to conventional chemotherapies. But I think the key feature going forward, as we don't know how long to continue these therapies for, and at least if you look at some of the IDELA data or some of the duvelisib data, there's a high discontinuation rate secondary to GI toxicity, that I think drugs that have an improved safety profile may offer new opportunities to allow patients to stay on therapy, which could translate theoretically down the road to much better efficacy if the drug has better safety associated with it.